Good afternoon for those of you that have connected in to the Aptamer Group presentation this afternoon. This is David Collins, the Chief Commercial here. I'll just say a few words whilst we wait for some of the stragglers to connect in. It's my job just to welcome you to this second Aptamer Group webinar in this series that we've planned. As I said in the first one, we've been overwhelmed by the response, and we have had more than 100 people registered globally. So I think that shows the broad range of interest that there is in this topic. We're doing, as you know, an integrated series of 3 presentations, and we've already covered Aptamer selection and the technology to do that. And that seminar was recorded and is available for reviewing. But now today we're going to start covering applications and uses of Aptamers. We are planning a 4th webinar in the first quarter of next year, which will focus on cell targets. But today we're going to do large molecule protein targets, and the next webinar in November will cover small molecule targets. The presenter for this series is Dr. David Bunker, our Chief Technical Officer, and he's a co-founder of the company. He's got 18 years experience of Aptamer science, and he emerged from academia to set up the company in 2008. And in his time in this field, he's selected more than 200 Aptamers against a very wide range of target types. At the end of this presentation, there'll be a question and answer session. If you want to ask some questions, there's a link on the YouTube page that this webinar is being broadcast on. And there's also a form on our website, excuse me. So you're able to submit written questions through those 2 routes. So I think we're ready to start. I will now hand over to David Bunker, the Chief Technical Officer, who will talk through applications of Aptamers selected against protein targets. Thank you.
Thanks, David. So yes, I'll be talking to you today largely about protein-based Aptamer selection and some of the applications, some of the things that you can do with an Aptamer against a protein-based target. So this slide's just as a reminder to say in part 1, we covered Aptamer selection in general, just loose terms, how we go about generating Aptamers. And as David mentioned, that presentation is available through our website. So anybody who wants to review it or use that for purposes, please feel free to take a look at our website. Okay, so this slide just is here to remind you of the services, how we go about working with customers and collaborators, the phases that we go through as part of our selection process. And this really helps us keep in touch with our customers and deliver in specific milestones, much as you would do on any other project type. So the first phase is our feasibility study. This is really there to assess how likely your target is to be successful in an Aptamer selection. Obviously, if we progress through that, we can move on to the polyclonal Aptamer selection. Here we can include counter selections. I'll come on to the importance of those later. Once we've generated our population of Aptamers, we move to phase 3 for this selectivity profiling. So that's just demonstrating how specific your Aptamer is and if it functions as you would intend. We then move on to the individual or monoclonal Aptamer isolation, and then we can finally move on to a minimal fragment identification and supply. The other parts to what we do, as well as developing the Aptamers, we can then help you to integrate those into various diagnostic devices. I'll be talking about that a little bit more in a moment. We can also work with protein-based targets for purification column development, and I'll come on to that as well. Biomarker discovery fits largely into the seminar we'll be doing next year on cell-based targets, but there is an element of protein-based selection to it, so I'll touch on it very briefly here as well. So just as a reminder, in its simplest terms, an Aptamer is a synthetic DNA or RNA molecule that's been selected to bind to a particular target. So in that regard, they're often thought of as the nucleic acid equivalent of antibodies. And I've trimmed my figure down from the previous presentation to this one because this is the focus of today's talk. So this is an aptamer recognizing a protein target, so the protein in orange, and the aptamer recognizing that protein in much the same way that an antibody does. So forming a complementary interaction with a given surface. And just to summarize again why you would want to use an aptamer, well, theoretically, aptamers can be generated against any type of target. So throughout the literature and throughout the experience in the company now, you can generate an aptamer against virtually anything. You can actually work with a lot of targets as well that are not amenable to other platforms. So antibodies, for example, it's very difficult to generate an antibody against something that is non-immunogenic, so it doesn't stimulate the immune system, or indeed is a toxic molecule. So obviously your animal would probably keel over and die before you got an antibody out of it. So we can work with that sort of target in our in vitro selection processes. We're also not limited to a biological environment, and that comes on to a lot of the applications I'm going to talk about today, where you might want to work in situations other than a nice neutral pH buffer. We can also tune the affinity and specificity of the target. So this is particularly important when it comes to things like protein purification and ELISAs, Western blots, that sort of thing, where you might want to build in or build out cross-reactivity with certain target types. So I'll come to those in a moment. The most important point from a lot of these applications, though, is the final point on this slide, the time to development and the fact that they are in vitro manufactured through well-known solid phase synthesis approaches. So this means that we can make large amounts of aptamers, up to kilograms if necessary. This gives us the ability to introduce very strict quality control. So you have the ability to produce GMP or CGMP grade aptamers if necessary. But it also means that you have much, much lower batch-to-batch variability, meaning re-optimization and revalidation of assays when you get a new batch is a lot easier. And if you get a bigger batch, you have to do that less frequently. So I'll come on to some of those points in a moment. And just to remind on the selection process itself, so we essentially run this cyclic iterative process to generate aptamers that are specific and high affinity to your target. I'll come back to this at different points throughout the presentation. It's just there to serve as a reminder to say we can build in different criteria at different stages of this process. So for example, in protein purification, you might want to build in a defined binding buffer where the aptamer binds to the target, and then a defined elution buffer where it lets go again. All of this is possible because we have an in vitro process where we can control the conditions at every step in this cycle. Okay, so the remainder of the presentation is going to be focused on developing aptamers for a particular application and their integration in those assay types. So I'll come on to those and mention them as we go. So the first point with aptamer synthesis, as I mentioned, it's largely by solid phase synthesis, which means you have a lot of control over exactly how you make the aptamer. And that can include the addition of functional groups for addition of other reactive groups such as fluorophores or gold nanoparticles, or even attachment to other substrates like a column resin or a sensor surface. All of these are possible through very, very simple chemistry. So for example, you could attach a thiol group to the end of the aptamer and then react any number of things with a compatible maleimide group. So I'll come back to those as we go through the presentation where you might be using them. Another point that I touched on earlier is the specificity that's possible with an aptamer through careful control of the selection. So the data presented here is quite old data now, but it just demonstrates how specific an aptamer can be. So on the left, we see some SPR data generated using an aptamer that's capable of recognizing 3 different conformations of the same protein. So you've got a cross-reactivity almost there with different folds of this protein. However, the aptamer on the right-hand side is actually able to discriminate between those folds. So what's important to point out here is that the green protein that is not recognized at the bottom by this aptamer is not different in terms of its sequence, its post-translational modifications. There's no mutations in that at all. It's purely a folding difference. And the aptamers, you can see, very clearly discriminate between them. So that just gives you an idea of the sort of differences we can recognize. Obviously, traditional differences as well, like post-translational modifications, methylation, phosphorylation, so on and so forth, you can discriminate those as well. So when it comes to assay development, there are a number of advantages to using aptamers that I'll just briefly summarize in this slide. So I've touched on the tunable affinity and specificity earlier. The other point, as I mentioned, is how readily an aptamer can be modified. So you could say that an aptamer is much more uniform in its labeling. So for example, if you were working with an antibody, you might use, let's say, an amine reactive dye, in which case you would get dyes at different points on that antibody. You can't really control that as readily. Whereas an aptamer, you could say, I've put a thiol on the 5' end, so I know that each of my aptamers is only ever going to be labeled at the 5' end. So that's the site-specific labeling that I'm mentioning there. It's also important to remember that that single point might serve as a point of attachment to, for example, an ELISA plate or a gold nanoparticle. So in that situation, you can say for certainty that all of the aptamers that are attached to your gold nanoparticles are all in the same functional orientation. Now, this is a problem for a lot of other technologies where, for example, if you are attaching an antibody to an ELISA plate, you might find that 50% of your antibodies are lying on that plate surface in a non-functional state where the antigen binding domain is actually face down, shall we say. So not, even though they're in the plate, they're not available for binding. So you've got a much higher surface capacity with an aptamer. You can use aptamers in tandem with other technologies as well. So we mentioned antibodies there. A lot of times we get approached saying, well, we have an antibody against our target already, but we need to develop a sandwich pair for our assay. So we can actually work with that and develop an aptamer that works in tandem with your existing antibodies. So this is helping a lot of people to overcome that requirement for a secondary antibody. As you can see in the little diagram at the bottom there, aptamers are also quite a bit smaller than traditional antibodies. Even antibody fragments are generally slightly larger than aptamers. This means that you can have a much higher packing density on the surface of a nanoparticle or a column or an ELISA plate. So you have much more surface available for reaction with your target. The aptamers are also less prone to aggregation at high concentrations. This can be problematic for certain affinity ligands like antibodies. So just a few of the advantages there. So to come on to some real-world applications. So I absolutely love this phrase, the ELONA, the enzyme-linked oligonucleotide assay, is essentially the aptamer equivalent of an ELISA. And you can see there that they work in exactly the same way. So essentially, the antibody has just been replaced by an aptamer. The rest of the assay can be exactly the same. So in the example shown here, we've attached a biotin group to the end of the aptamer in much the same way that you would with an antibody. And then that allows you to use strep HRP and TMB substrates, so on and so forth, exactly the same as you would do with an antibody-based assay. So this is what we were referring to previously about simple conversion of one assay type to accommodate an aptamer. So it's an example here. You could go a little more advanced if you wanted to and replace the strep HRP, TMB substrates and so on by including a fluorophore on the end of the aptamer and just have it as a direct detection. So it's a bit more sensitive, but this way is a lot more closely related to what people are used to with ELISAs. The next slide is just talking again about aptamer-based use in lateral flow devices. So as I mentioned earlier, you can attach aptamers quite readily to commercially available nanoparticles. These can be gold or latex or any other number of particle types with compatible functional groups on their surfaces. So you can see there a very simple cartoon just demonstrating how this works. So the aptamer would essentially be conjugated to the gold nanoparticles, and as the analyte is applied to the sensor strip, you then get migration of the material through capillary action, and where the aptamer has recognized its target, you get a buildup of a stripe forming what's referred to as a test line, and then a control line is possible. And this is very easy with aptamers because you can simply build that up as a complementary oligo sequence. So the control line is much easier to work with as well. So on the right-hand side there is just an example of a very crude, very early lateral flow test that we did with an aptamer of ours, showing how well you can conjugate that to a gold nanoparticle, showing that there's no recognition of this non-target protein, but there's a very clear spot forming around the target protein site. A step on from there is a very familiar technique of Western blotting or spot blotting. So here, again, aptamers can readily be just converted and used in this sort of format. So where you would wash the membrane post-transfer with an antibody solution, you would just do exactly the same in the situation with an aptamer. And again, you can use any number of reporter groups on there. So the situation on the left-hand side there, you can see a concentration-dependent binding series using a fluorescently labeled aptamer, whereas the situation on the right, you see concentration-dependent binding with a radioactive labeled aptamer. So two different formats for the same assay type, and you can see the sort of sensitivity you can get there. But also what's important to note from the right-hand side data is the aptamer there is able to recognize its target in the third, fourth, and fifth situations in crude samples, so ex vivo material. Without any purification at all, the aptamer is still able to recognize its target. So they're very, very useful in this format. We also have issues of cross-reactivity is a common problem that we're asked to address. So people come to us and say, well, my antibody recognizes several different targets on my blot, which is making data interpretation difficult. So we can build that into the aptamer selection process. We can even build in a step where we generate aptamers as they function in a Western blot. So that's something we can do as well. So this next slide is going a little more advanced, using aptamers to develop biosensors. So in this case, a simple dip and read sensor. So in this situation, it's very common to characterize an aptamer, how well an aptamer binds to its target, how specific it is, so on and so forth, using systems like biolayer interferometry or SPR, surface plasmon resonance. In those situations, typically the target, the protein, is attached to the sensor surface, and then a trace is recorded when the aptamer interacts with that surface. But it's very, very easy to envisage flipping that situation on its head and immobilizing the aptamer and doing the same thing, studying the interactions that way round. But what you can see there is if you do that, you've instantly turned that sensor from a generic platform into a target-specific platform. So you've created a biosensor out of a generic platform. So just as an example of that, in the data in the middle of the slide here, you can see an aptamer recognizing the target protein in green with quite a high affinity, but not recognizing any of these non-target proteins shown as almost baselines in orange and red there. So you can convert a generic platform into a target-specific one. And again, just to expand on that, so for example, in the Blitz system from Forte Bio, something we work with, you can take their generic platforms, their generic probes with streptavidin or thiol or amine reactive probes, attach an aptamer to the surface and make a specific biosensor for one sample in this case, or with some of the higher grade instruments, multiple different analytes in parallel. So you can really use this to develop different biosensors. An extension of that is electrochemical sensors. So in this situation, you essentially attach the aptamer to a sensor surface, represented here by the little yellow rectangles. So there's your sensor surface with an aptamer attached to that and a redox reporter. So any sort of sensor redox reporter there is possible. And what you see is when the target interacts with the aptamer, quite often you will get a conformational change, which changes the distance between the redox reporter and the sensor and gives you an impedance change or some sort of readout based on that sensor. So the sort of data you can see at the bottom there. So that's sensor-based applications. Very different from that, but using the same sort of principles, is protein purification. So as we call it, AptaBind. So in this situation, as well as generating an aptamer that recognizes your target, you have additional criteria here. So not just high affinity and specificity, but you also want release conditions built into these selections as well. So you can see at the little cartoon at the bottom left there how protein purification works. You essentially load a crude matrix onto the column in the loading step, wash away anything that the aptamer doesn't recognize in the second step in the middle, and then specifically elute the recovered material and essentially partition the crude remaining material from the purified target ligand. So we can do that as built into each of the different steps of our selection process. The loading would be your interaction phase. We can then build in a wash step for the separation and then target specific elution. The point with this is that we actually have a lot of control over that elution step as well. So this is very important for rare proteins or fragile proteins where you might not necessarily be able to employ a harsh elution condition, like let's say urea or very high salts that might be required to elute from other affinity ligands like antibodies. So up at the top right-hand side there, you can see an example of this, again, monitored on a biolayer interferometry instrument, but the concept is the same as you would have in a column. So the aptamer is first loaded onto the sensor surface in this case, using the same chemistry that we would attach it to a column resin, so an amine reactive group, for example, or a thiol group. You can see in the next phase that there is no interaction with the buffer at all, so the trace remains flat. But as soon as we introduce a target matrix containing the protein of interest, you see we get a very clear response that remains constant. There's a slight drift throughout the wash phase, which is what you would expect. And then as the elution phase is introduced, the target elution buffer, you can see that the protein essentially drops back off the sensor surface. What's also important for column development is the ease of reuse. So you would want to be able to use an aptamer column multiple times to purify your protein so that you're essentially not having a single use column. So we can also build in that criteria to say, for this aptamer to be useful, we need to be able to load it, wash it, elute it, and then repeat that cycle over and over. So we can build that in using standard regeneration conditions, which I'll talk about later. So just an example of how well that can work, we have a situation here where we've used an aptamer-based column to demonstrate purification of a customer target. And you can see there from the input material, which is quite crude on the left-hand side next to the marker, to elution band 1, 2, and 3, you can see there that the target is almost 95% pure through a single purification pass. So very, very specific targets using very specific elution conditions. Now, in this case, the target protein was quite valuable, and the customer here was losing a lot of material due to the nature of the elutions that they had to use previously. So we wanted to engineer here a much more gentle elution condition. And what I mean by that is, for example, we could generate an aptamer that is responsive to a small molecule. So introduction of that small molecule would change something about the protein or the aptamer and make them release. Or, for example, you could adjust the pH slightly. So changing the pH resulting in elution of the protein. So we can engineer those in depending on the nature of your protein and what you need to work with. So I mentioned right at the start briefly about biomarker discovery. So this is more of a cell-based application, but I'll touch on it here. So as you can imagine, cells quite often express different proteins on their surface, depending on whether they're healthy, their location, whether they're diseased, so on and so forth. So in this case, we've just demonstrated this simply using a red rectangle. So we don't need to know what that target is. We just need to know that it's important to the customer to say, we need to know what that red rectangle is. Can we identify it? So we can actually use a whole cell as a selection target and generate aptamers that are specific to that red rectangle by using a cell line that doesn't have it as a counter selection target. So using our cyclic process that we mentioned right at the start, we generate aptamers that recognize that, and then using the AptaBind approach, we can then take those aptamers and attach them to a solid support and use them to purify whatever that red rectangle happens to be. So essentially using that aptamer in a immunoprecipitation-like assay. So the point to note here is that we don't need to know anything about the nature of that target other than it exists. An extension of that, again, slightly more cell and tissue based, but it's applicable here. So we can generate aptamers that recognize protein targets and then assess them for the ability to recognize that target as it exists in a paraffin embedded tissue sample, for example. So an example here, this is not some of our data, but you can see here quite clearly the aptamer comparison with a commercially available antibody generated against the same target. So again, this comes down to our in vitro process. Because we can generate the aptamers against the protein as it would be presented in a formalin fixed sample, we actually get aptamers that are fit for end purpose. So we start with the end in mind. So in conclusion, aptamers function as nucleic acid equivalent of antibodies. They're selected under defined conditions with your end procedure in mind, and we can modify them to give them a whole range of different functionalities. And because of that, they're readily adapted and amenable to development in research tools, diagnostic tools, and, as we'll touch on later, therapeutics. I'll thank you for your time and attention, and we'll take any questions that come through, and we've got a few already. So, David, if you can read those out for me.
Thank you very much, David. Hopefully that was of interest, and we have had some questions here. We will try and work our way through them all. If we don't, we will follow up by email, but we'll try and answer them all in the time available. So we're in good time. So we've got 10-15 minutes of question time if needed. This is a really good one that we've received. Can one aptamer be used for multiple applications? So could you use the same aptamer in a Western blot and for purification and for an ELISA?
Absolutely, yes. So that's one thing that I enjoy saying to people when we go out to meet customers. It's pretty straightforward if you take an aptamer, put a thiol group on the end of it, you can have different people using that same aptamer for different applications. Obviously, there's going to be some limitation on that. So if the protein conformation is different in an FFPE sample, for example, you might not be able to use an aptamer developed for an LFD. But apart from that, yes, you should be able to use the same aptamer in multiple situations.
Focusing on the AptaBind application there, we've got quite a specific technical question here. What elution conditions are available for affinity columns? Does the anchor affect binding?
Okay, yeah. Yeah, so the elution conditions really come down to customer requirements. So, if you need a gentle elution condition, we could, as I mentioned, engineer in a requirement for the selections, let's say magnesium. So we could say that the aptamer requires magnesium to bind, and then we can strip that magnesium away with something like EDTA. Without that magnesium, the aptamer no longer functions and the target is released. So you can engineer those conditions quite carefully. with regards to the linker chemistry, yes, you would have to be careful as to the type of chemistry you use, but a lot of the time you would immobilize the aptamer with an irreversible linker, so click chemistry, for example, or amine NHS coupling, so something that's not going to be affected by your end dilution condition.
Thank you very much. There's a question as well about, we're back to the sort of comparison and utility against antibodies. If I use antibodies for my Western blot, is there any reason that I should use or switch to Aptamers?
I would actually say that's down to you. If you have an antibody that works in your assay already and you're happy with it, carry on using it. That's absolutely fine. What I would say to customers is, if you have something where the antibody works but it's not particularly great, or you get cross-reactivity, then maybe consider switching to an aptamer. As I mentioned earlier in the talk, there's all the specificity advantages for aptamers. We can also work with them under denaturing conditions if necessary. So we know that a lot of the times protein gels are run under denaturing conditions, so an antibody might be generated against the folded form of the protein and not recognize it as it is presented in a Western blot. So as I mentioned, we can build that into our selections so that the aptamer recognizes that conformation.
Someone who's online is an aptamer selection scientist themselves, and they're performing selections as well. And this is quite a practical technical problem. They've noticed that they barely get about 1% bound until generation 5. I think we would use the term selection round 5. Mm-hmm. And then spiked in 6 before dropping again for selection round 7. Is there any way that he or she can improve binding?
Okay, I'm just reading through that question.
There seems to be some fluctuation from round to round, and there's a drop after quite a high level there. Mm. Are there any thoughts about that?
Without looking into your selection method in too much detail, I couldn't say for certain, but I would be willing to guess at that point you've introduced a counter selection or some other selection pressure, and that's knocked a lot of your population out. What you will probably find, if that is the case, is that your generation 7 population is actually more specific, even though you've got less of it. So without knowing a bit more about your selection process, I can't say for certain, but that would be my guess.
And perhaps a practical piece of advice here, which may be being done, is that from each round you should split that, keep an archive sample, and then if you do progress forward unsuccessfully in the next round, you can go back to the previous round, use that sample, and maybe do the selection under different conditions or something like that.
Yeah, that's possible.
Okay. Have we got any other questions in? No, I think we've covered everything there, Dave.
Okay.
Okay. So it just remains for me to thank everyone that's dialed in for their time. We hope that was interesting and useful. It was great to see some very specific and technical questions and people involved in aptamer selections themselves. This webinar, as I said at the beginning, has been recorded, so you can watch it again or refer your colleagues to it for viewing, and the previous webinar. I want to thank my colleague who's here in the room with us, Joel Sutcliffe, who set this up. He's done all the administration and will contact you again soon with information about the next webinar. That's on the 29th of November, and it will focus on applications for aptamers against small molecule targets. So there's some very specific aspects to that Dave Bunker will cover in that seminar. So if there's any further questions or project-specific inquiries, we're available on info@aptamergroup.co.uk. Just remains for me to say thank you once again. Enjoy the rest of the day, and we hope that you'll join us next time. Thank you very much. Goodbye.